You were looking at images of New Orleans. We are at AHA 16. You know, this was an aspirational term just 10 years ago, but genomic medicine is already impacting a variety of clinical scenarios by an increasing number of healthcare professionals, and it is finally coming to cardiology. So in the December 27th issue of JACC, a great way to end 2016, is with a state-of-the-art review that discusses proteomics research in cardiovascular medicine and biomarker discovery. And to do that, I am with the authors of the paper. In the middle is Elizabeth Murphy, who is a PhD and a senior investigator at the Laboratory of Cardiac Physiology at the National Heart, Lung, and Blood Institute. And far away from me, we have Maggie Lamb, who is a PhD and an assistant project scientist for the NIH Center of Excellence in Biomedical Computing at UCLA. And also right here next to me is Peipei Ping, who is a PhD also and a professor of physiology, medicine, cardiology, and bioinformatics at the David Geffen School of Medicine at UCLA. Before we discuss proteomics in cardiovascular research, can we talk a little bit about where it's being successfully applied elsewhere in medicine? Who wants to start with that?
Definitely cancer. So the NCI has an initiative, has a consortium that's called CPTAC, Clinical Proteomic Tumor Analysis Consortium, that's been very, very successful in identifying markers for tumors and to classify tumor grades. And that, I think newly there's a push to actually look at the the functional consequence of mutations in tumors, and how do the genetic variants contribute to changing protein functions and so on.
I agree entirely. Just follow up what Maggie said. NCI is one of the other large NIH agencies that has launched a substantial amount of efforts and resources using proteomics to create biosignatures to discern over 157 different cancer or tumor cell lines. Wow. And they've had tremendous success in the past 15 years. Indeed, proteomics technologies has become one of the driving tools for cancer biomarker discovery in that particular field. And add to what Maggie has said, there are two other agencies that have also launched similar efforts. One is National Institute of Aging, and they have both intramural and extramural programs using proteomics technologies to tackle different questions in aging-affiliated symptoms, including neuronal disorders. The other agency that has spent considerable amount of efforts in this area is National Institute of General Medicine, NIGMS, has funded a number of programs supporting both the bioinformatics structure of proteomics as well as a specific structure biology program. It's called Protein Data Bank that collects over 4,500 crystals, structured images of different proteins, in particularly memory proteins, which are primary drug targets in many diseases.
Okay, so let's talk about cardiology a little bit. Over the past couple of decades, there has really been this accelerating growth in the application of proteomics in cardiovascular biomedicine. What have we learned so far?
Well, I think we've learned in two areas, I would say. One is in more of a basic area. I think it's become very clear that when they sequenced the human genome, that there weren't as many mRNA, there weren't as many genes as they thought there were going to be. And so a lot of the complexity that makes us makes humans unique is that there are all these post-translational modifications that are. So you have one protein, but you can have many different marks on it. You can have phosphorylation, S-nitrosation, oxidation, acetylation. So there are many different modifications on it that make the protein behave differently or target differently in the cell. And proteomics is one of the main tools that we're using to discover how this variation occurs and how all this different signaling occurs. So I think that's one basic and very powerful basic tool that's being used. In addition, which we've already started to talk about, biomarkers. And I think that, that cardiovascular sciences are coming along, and that there are, at this meeting alone, I've heard many talks about people identifying potential biomarkers that could be used for heart failure or ischemic heart disease. And so I think it's a very emerging area, and proteomics is a main tool being used to study these proteins in the blood and serum.
So what are you trying to do in this particular paper? It is a great review. I've read it. What are you hoping to help people understand?
Well, mostly that proteomics is accessible, and that with proteomics we can learn a lot about the disease mechanism and discover disease signatures for potential biomarkers. And there are great opportunities with proteomics that we can know a lot more about the cardiac proteome. So proteomics is great.
In terms of how it will affect research, this biomarker activity will obviously help. Is there any other aspect of research that may change with what we find in proteomics?
So as Dr. Murphy was just saying that we can look closely into what are the post-translational modification, and there are other areas of proteomics, such as we can look at protein-protein interactions, protein localization, or protein turnover. These are parameters of the cell and of the proteome that we cannot discern from just using genomics tools and other tools. And
So are we heading towards more of a individualized medicine? That's where everybody keeps promising us. We haven't gotten there yet. Is this going to get us there?
I think it will help get us there, because I think that, plus there are different splice variants in the cells, and so we can pick these up with proteomics as well. So I think that different patients with different types of disease may have different markers in their cells and in their serum and plasma that we can pick up with proteomics in the future. At least that's a future goal.
So I agree entirely with both Tish and Maggie. So when we look at information we have gained from genomics and genetic studies, as well as transcriptomic studies, we begin to understand as individual human, the differences of my genome versus that of Dr. Murphy or Dr. Maggie Lam are actually minute. People started with different transcriptomic studies and variance analysis found some differences, but really we're looking at the human proteome, the cell proteome, to look for additional unique features that are capable to distinguish each one of us as a unique individual. And that's where, as printed out, the unique characterizations of post-translational modifications, by the way, there are 650 of them, different types, that would give us a completely different signature profiles to say, that's where Dr. Maggie Lam is, that's where Dr. Tish Murphy is, that's why they are unique, who they are. But if you look at the genomes, 99% of us are identical.
We're a lot closer to each other than sometimes
That we thought we were.
Exactly. Starting with you, Maggie, some example of what you're working on that kind of give us a concrete example of the work that's being done. What are you working on right now?
So I'm working on, so as you know, there are 20,000 genes, protein coding genes in the human genome, and you can create up to about 100,000 protein isoforms. So one way to create this proteome complexity is through alternative splicing. So traditionally, the study of alternative splicing at the protein level is very restricted to single target study because of multiple technological challenges. So I'm working on developing a strategy to help to look at protein alternative isoform at the proteome level, at the proteomics level, so that we can know more about how protein isoforms contribute to the possibly cardiac disease phenotypes, and certainly how the proteome, alternatively spliced proteome, is being impacted by disease.
I'm working primarily looking at post-translational modifications and how they might influence disease. But one thing that I want to follow up on what Peipei said, and that is, if you look at, people have done a lot of sequencing and looking at changes in the genome, but what really matters for how our organs respond and how heart responds is the proteins. And there was a very nice paper from someone from Peipei's lab in one of the Young Investigator competitions showing that there's not a very good correlation between changes in protein and changes in messenger RNA. And it's really the protein that matters. And so not only do we have to know what the protein changes are, and that's something that I'm interested in, how do proteins change in heart failure? How do they change in ischemic heart disease? And what are the modifications of them that influence their activity?
So I'm interested in dynamics of the cells and how not just cell live and how they respond to therapy. And so the temporal response of proteins to drugs and to other stimulus is a primary interest of our laboratory. And the other part of it is we're interested in building computational tools that could help analyze the signatures and biomarkers that we gain from these experiments and to discern phenotypes and to identify specific unique sets of signatures or targets that could have some predictive power for the phenotypes to either transition response to therapy or transition from pathogenesis, restores function to health.
Am I correct in assuming this is an exciting time for you guys in research?
Very much so. The tools we have now are just phenomenal, and I think we're ready for an explosion in this field of new knowledge.
Well, you need some of the new knowledge, and so for the end of the year, why don't you check out the JACC December 27th issue? That's where this review is from. And please check out the rest of our coverage from AHA 2016 for CardioSource World News and CardioSource World News Interventions. I'm Rick McGuire.